time PCR. So some of you have done PCR, some of you are going to be doing PCR this week. So just as a reminder, since you've all had the basic PCR lecture, that when you are doing a polymerase chain reaction, you are adding in the template, you're adding in the oligonucleotides that are going to bind on either side of your target sequence on the templates, on the two DNA strands. And you are adding nucleotides and an appropriate buffer so that you have a good pH. And you are adding in Taq polymerase, Thermus aquaticus polymerase, which is specifically designed to work at 72 degrees, a high temperature. And the three steps of PCR are denaturation of the two strands of DNA, annealing of the oligonucleotides to their target sequence, and extension by the Taq polymerase of in a 5' to 3' direction of both of those primers. And you do need a primer to be able to initiate the DNA sequence addition with Taq polymerase. And also, as a reminder, the denaturation is at a very high temperature, usually in the 90 to 94 degree range, so that you can make sure you're completely denaturing the DNA. The annealing temperature is going to be determined by the sequence of the oligonucleotides that you are binding. The longer the oligonucleotides, or the higher the GC content, the higher the melting temperature will be. And so the annealing temperature is usually about 5 degrees below the melting temperature. And also, remember that I told you during your lecture that if you order an oligonucleotide from a company, such as IDT, which is up here in Carlsbad, or has a branch in Carlsbad, on the paperwork they send you, they will tell you what the melting temperature is, so you don't need to worry about calculating it, although there are formulas that you can find online if you needed to do that. So denaturation, annealing, and extension. Since Taq polymerase works best at 72 degrees, but does not denature too fast at the 90 degree area, it became a much better choice for PCR than a regular polymerase, regular DNA, Pol2 or something like that, which works best at 37 degrees, but by the time you're up to the 90 degrees, it will denature pretty quickly. Now, this is not to say that it can go on forever. So remember that you have to have all these components, all the proper components in your solution. And when you run out of the proper components, then you're no longer able to double the number of your target sequence each time. So this should all be very familiar based on the previous lecture to you. So this is theoretically what it's going to look like. You'd have, based on your target sequence, you go, even if you started off, say, at 1 copy, then you do 2 and 4 and 8, etc. So you double and double and double and double until you run out of something. And when you run out of something, whether your polymerase dies through, because remember, enzymes have a half-life, and so the longer it spends at 90 to 94 degrees, the less time you're going to have out of it. And or you might run out of nucleotides, or you might run out of something. So so it will double and double until it runs out of something, and then it will stay at the same concentration. But every sample might run out at a different point. So when you're doing this is called endpoint PCR. So you're not watching how the PCR is happening until the very end. When you take your sample, like we did, we got the result of our PCR, and at the end of our PCR reaction, we ran it on a gel to see how it worked. And you guys saw on the gel, everyone had potentially different concentrations of the target sequence. So it might have been because maybe a little more template went in, or maybe in your particular sample you had a little bit less enzyme, so the total enzyme died out sooner, etc. So You don't know exactly how much template you had in there if all you're doing is looking at the end result of the PCR. So, ways needed to be addressed to come up with a way to try to measure how the reaction is going before it actually finished, before it ran out of something. And so this is the idea behind real-time PCR. It measures in real time what's happening. Now, a big advantage in real-time PCR, what you did in class or are going to do in class is completing the PCR reaction, waiting for all the cycles to finish, and at the end of the cycle finishing, then you're running the gel and staining the gel and taking a picture of the gel. All of this takes time. So if there was a way to monitor what was happening in the tube while it was going on, this is a big advance, or this was a big advance when people came up with this idea first, so that they no longer have to run it in a gel, and they no longer have to wait until the end of the reaction to see what's going on. So basically they can pick a point maybe halfway into the reaction and say, okay, you know, how much sample are we going to have? So basically so this is like a theoretical idea, and theoretically there's a baseline below which you're not going to be able to detect the target sequence being made until you achieve a certain number of copies. So even though it looks like there's high here, that's actually just the nucleotides it's measuring, and so it'll be approximately the same for a while until you're able to start measuring the amount of target that's being made, and you want to choose your point that you're going to measure before it starts leveling off due to running out of one of the components in the solution. So you want to get it while it's still in basically logarithmic growth phase, just like you did with the bacteria. So there are different ways that people have come up to do this. So this is an example, again, of why this might be needed. So this is just a series of 10-fold dilutions. So this one was the most concentrated DNA, and they diluted it 10 times, diluted it 10 times, diluted it 10 times, etc. So you can see that even though they all start off sort of in the same range, they run out of things in the reaction at very different places. So, while this one had the highest DNA concentration, this one, which is 10 and 10 and 10-fold diluted, actually had a higher yield of product. So if you were just running all these on a gel, you would not get an accurate idea of if these were, say, different things that you were trying to amplify, you would not get an accurate idea based on this endpoint PCR analysis of maybe, which ones were in actual higher concentration in a cell. So doing real-time PCR enables you to pick a point in your reaction while everything is still very logarithmically growing and get a much more accurate idea of what there is in the sample. Additionally, this is used in things like military applications. So if you have battlefield conditions where you suspect a particular, something like anthrax is present, you can have a PCR setup where you can have the correct primers, the Taq polymerase, and you can just take a scoopful of dirt out of there. Maybe you might need to amplify the DNA. You might need to purify the DNA out of it sometimes. And there's probably a bunch of different bacteria, including potentially anthrax in the dirt. But you're going to have anthrax-specific primers in your setup. And by having that, it's going to amplify the targeted gene that might only be found in anthrax. And because you don't have to wait for the endpoint, you don't have to run it in a gel, you can see very, very quickly whether you have anthrax present under battlefield conditions. If you suspect that people are getting sick and maybe chemical weapons have been used. So there are a number of different types of bacteria you can do this for, not just anthrax. But the military actually developed real-time PCR, and then it found its application in life science research for people being able to say, hey, sure, they developed it for this, and then DARPA is great in terms of funding all kinds of research that later find real world other than military applications in life sciences. So biotech companies said, hey, yeah, we can use it for this, we can use it for that, stuff like that. Okay, so one of the things that they use is called cyber green. So cyber green is a chemical, much like ethidium bromide, that's going to bind to double-stranded DNA. So you're making your PCR reaction, and when you're making your PCR reaction, you just throw in like a microliter of a solution of cyber green. And the more double-stranded DNA is present, the more cyber green is intercalating into this double-stranded DNA. And when you hit it with a particular wavelength of light, much in the way you saw with ethidium bromide, where you hit it with an ultraviolet wavelength, and it put out a visible wavelength. So much in the same way, you have cyber green, you hit it with a particular wavelength of light, and it puts out a different measurable wavelength of light. So the more double-stranded DNA you have with cyber green, the more particular wavelength of light is going to be detected. And if you don't have double-stranded DNA, you're not going to get that particular wavelength of light. So it's pretty simple to use. Cyber green is pretty simple to use. So there are advantages and there are disadvantages to using cyber green. So the advantages is that it's inexpensive. You can just buy a bottle of the chemical and boom, you add it to the PCR reaction. It's easy to use. So there's no design or optimization of primers or anything like that that's needed to be able to use it. As I said, you just plunk it on in there, and it's fairly sensitive. So it's able to fairly accurately represent how much double-stranded DNA you have. The more you have, the more of that particular wavelength of light you're going to get. But having said that, in anything in biotech, there is no magic bullet that works all the time. So there are also some disadvantages to using cyber green. So the disadvantages, it will bind to any double-stranded DNA. So if you have primers, and we talked about needing a hot start DNA to reduce non-specific priming of areas of the DNA. But if you have some non-specific priming going on in your sample, it's going to bind to that double-stranded DNA, so it cannot distinguish between a 100% correct target and mispriming events that happen in the DNA. It also cannot distinguish between what's called primer dimers. So if you have primers that have overlap, a little bit of overlap on the 3' end. So if you have one primer, say right here, and let's say this primer happens to end in a GT, and you have another primer right here, this primer happens to end in an AC, just for the heck of it. When you mix those two primers together in solution, the 3' ends are going to bind to each other during the annealing phase. And then when polymerase comes along, it's going to extend this, it's going to extend that, and boom, you get something called a primer dimer that's double-stranded DNA. Now, when you are when you're designing your primers with a company, there are programs that you use to say, how much of a possibility is this? Should I worry about this? And so the programs can alert you to the fact that this is going to be a huge problem or shouldn't be a big problem, and then you can say, okay, do I need to change the sequence of my primers and stuff like that. So if you have primers that can bind to each other and make double-stranded DNA, even though it's completely not your target at all, the cyber green is going to be able to bind to your double-stranded DNA. Okay, and as in this case, non-specific reaction products. It's difficult to use in multiplexing. So remember, multiplexing we learned about in terms of the short tandem repeats, where we had in our PCR we had, or for half of you will have, 3 different PCR reactions going on at the same time in the same tube. The police department, remember, does around 24 reactions all at one time going on at the same time in the same tube, although with different tags on the primers, so that maybe each set of 4 different sizes can be distinguished one from another. But if you don't have some specific tags on your primer to distinguish them, and you want to do multiplexing in a tube, it's going to say, yes, you have double-stranded DNA, but until you actually run it on a gel, maybe to see what's going on in the tube, you won't know whether it's, which one of your targets. Maybe one target has a whole bunch and one target is missing completely. So that's less than ideal, because now you find yourself running a gel just like you would with an endpoint PCR. So the advantage to this is gone unless you really, really need to know what's happening while you are multiplexing. But having said that, there are ways to use it. So it's not impossible to use in multiplexing, it's just difficult to use in multiplexing. At the end of my lecture, I'll give you an example of how you can distinguish different targets in multiplexing. Okay, so at high concentrations can inhibit PCR reactions. But having said all that, if you're just looking to quickly ascertain whether you have a single or maybe even only 2 that you can distinguish through melting temperatures, maybe 1 or 2 targets, real-time PCR is an excellent type thing to use in your experimentation. But again, there's no one magic way of doing it, so different companies come up with different ways of doing things. One company invents cyber green and puts a patent on it, so no other company is allowed to use it with their technology unless they license it, and the licensing fee might be pretty hefty, so they're like, we're going to spend a little bit of money and find a way around this. So companies have found a different way of doing things. So this is just another showing you how it works. So here you'd have a strand of DNA, here you'd have a primer. When it's single-stranded, you'd have the cyber green in solution. As the polymerase extends, you have more and more of that cyber green now interpolating in the DNA, and when it's queried in a machine with a particular wavelength, now it's going to put out that visible wavelength. Okay, so another way of doing this. There we go. Another way of doing this is using TACMAN. TACMAN is a very specific polymerase that is used that has a 5' to 3' exonuclease in it, and it's used in conjunction with an extra primer that is specific for the sequence that you are trying to amplify. So here is your regular 5' to 3' and 5' to 3' template strands. And these are the oligonucleotides right here and right here that are binding to each side of your two DNA strands that you want to amplify. In the middle of the sequence that you are trying to amplify, you're going to make another primer. You're going to have a company make another primer that is very specific to the sequence that you are looking for. So now this addresses one of the problems with Cyber Green is you can use it in multiplexing and it's specific for the target. So you can tell, am I making more of my specific target that I'm looking for, and can I use it with multiple different reactions at the same time? Because every different reaction is going to have a different primer bound to it in the center of it. Each individual primer now can be distinguished on the basis of little tags that are put on both sides of the primer. And so there is a tag on the 5' end of the specific primer that's called a reporter, and there's a tag on the 3' end that's called a quencher. And when the reporter and the quencher are close to each other, within a certain distance of each other, they are going to absorb the energy. So the reporter can absorb the energy of a particular wavelength of light and transfer it to the quencher, and no corresponding different wavelength of light is emitted. So this says my primer is still whole and complete and intact. But what happens is, and during the annealing step, this primer is, in addition to the primer right here and right here, you have this extra primer, and it's also binding to its target sequence. And the polymerase that you are using in this case is called a TACMAN polymerase. And as I said, this TACMAN polymerase has a 5' to 3' exonuclease function, which means when it runs up against this extra piece of DNA, it starts chewing it up. Okay, so it's an exonuclease, so it says you're gone, you're gone, you're gone, you're gone, you're gone, and it just chews that into individual back into individual nucleotides, and then the reporter and the quencher are separated from one another. So when the polymerization, the double-stranded polymerization step, the extension step is completed, you basically have chewed up your primer, and the reporter and the quencher are now separate. And you might think, well, in a tube, there's going to be plenty of reporters and plenty of quenchers, and they all must be quenching each other. But on a molecular level, they're actually quite far apart. So compared with how close they are on a nucleotide level, and when you have them in a test tube, even in a 25 microliter reaction, even with a fairly high concentration of this primer, on a molecular level, they're all quite far apart now. And when it's chewed up, the reporter and the quencher are now on a molecular level quite far away from each other. So when you then have, say, a 25 microliter tube that you hit with a particular wavelength of light, now the reporter is no longer near the quencher, so it's no longer transferring that energy. So it's fluorescence, resonance, energy transfer. It's called FRET. Fluorescence, resonance, energy transfer. And so when the reporter is no longer close to that quencher, FRET no longer happens. The energy is no longer transferred to the quencher molecule, and now the reporter is putting out that energy in the form of another wavelength that can be measured. And that says, okay, so we have some of this that is now apart. It's been chewed apart, and that means we have our target sequence that's been made in here. And so the more of the and remember, there's lots and lots of these. So for the second round of amplification, you get another set of this that's going to bind to that target sequence and get chewed up again. And each time you're going to get more and more chewed up again. And so your wavelength of light that's put out is going to be higher and higher with every round of amplification. So when you are using real-time PCR, by knowing what a wavelength of light is being put out by the reporter, you can then say, okay, for this specific reporter-quencher pair, I'm getting, a lot. Or maybe you have a different reporter-quencher pair on another gene, and you're not getting nearly as good results with that one. So then you can say, okay, well, this one's not putting out, this one's not amplifying as much. So you can actually measure the different levels that are happening in real time, which is real-time PCR. And so by having different reporter-quencher pairs, you can now do multiplexing. And there are a lot of reporter-quencher pairs that you can have. And the machine is capable, the real-time PCR equipment that you are using, you just tell it which reporter-quencher pairs you are using, and it's able to measure the wavelengths of all of those reporter-quencher pairs very quickly. So it's maybe doing this at the end of each round of amplification, and it quickly runs through all of the querying wavelengths that you've said you have in this pair, and it's quickly measuring the output. And it can do this in, say, a couple seconds. So it goes very quickly. It's wavelength answer, wavelength answer, wavelength answer, wavelength answer, etc. So just boom, and it gets numbers of how much of this you have. So remember, you get more of this at the end of every round of amplification. So your level goes up and up and up and up. It doesn't go down and up and down and up. It just keeps going up at the end of every round of amplification. Now this is different than cyber green, because remember, cyber green, you just have a certain amount of cyber green in there, and it's putting out a specific wavelength of light depending how much is in double-stranded DNA. So if you were to test it while it is in the annealing phase, while it's all double-stranded, say at the end of amplification, you would say, okay, I've got a lot of it. But then if you were to test it at the end of a denaturation phase, then denaturation, it's going to be back in solution, because at denaturation, it's no longer double-stranded DNA. So if you were measuring it, say, at an incorrect point in the amplification, you could be getting incorrect results. So that's why you also need to tell your piece of equipment what type of technology you are using, so it knows, okay, I need to measure it while it's double-stranded. I can't measure it while it's, after it's been, say, denatured. But if you were to measure this even in the denaturation step, because the reporter and the quencher are now apart from each other, it doesn't matter what point in the amplification you measure this. This is going up and up and up, but the cyber green in a reaction is going to say low, high, and then denaturation, and maybe you measure it annealing, and so each time it gets higher, but when it goes to denaturation, it's always going back down low, because you are making those single-stranded again, so cyber green is going to leave your double-stranded DNA. So that's why it's important for you to have to know that your equipment is able to use the technology that you are choosing, and that you choose it properly, you program in everything properly. Otherwise, it's going to potentially be measuring at an incorrect point. Okay, so as I just said, companies patent their ideas, and so TACMAN got patented. So then another company wanted in, and so they came up with yet another way of doing it. And so they came up, so Molecular Beacons came up with something similar, but different enough that it's patentable. So what they came up with is this is the target sequence for the primer to bind to right here. So you're still making a primer that's going to be specific to the sequence. So you can still do multiplexing with this method and have different reporter-quencher pairs that are very specific to what you're looking at for, say, 24 different things all at once. And this is the sequence right here that's specific to each target that you want to bind. But outside, so if this is the 5' end right here, 5' going through here all the way here to 3'. On the 5' end you still have a reporter. On the 3' end you still have a quencher. But what's different here is the sequence outside of the target sequence is designed to bind to itself. And so this uses reporter-quencher pairs that are designed to achieve FRET, the fluorescence resonance energy transfer, only when they are very, very close to each other. Closer than in the TACMAN methodology. So again, you have lots of different reporter-quencher pairs, all with slightly different characteristics, so you can choose what you're going to need. So for this methodology, you're using reporter-quencher pairs that have to be very, very close to each other, and you are not using TACMAN because TACMAN is patented by the company that made it, so there is no 5' to 3' exonuclease activity when you are using molecular beacons. But what happens is when you denature, everything becomes single-stranded, and when you anneal, now the specific sequence right here is going to anneal to the target sequence, and now the reporter and the quencher are going to be far away from one another. And when the reporter and the quencher are far away from one another, you query the reporter, and now it's not close enough to do the energy transfer anymore. So now the reporter is going to be putting off specific wavelengths of light that can be measured. So this says, okay, during the annealing step right here, you can be asking during the annealing step, and so you would want to do this one before you start extending the other primers. Because remember, it's double-stranded. There's still going to be a primer over here. There's still going to be a primer right here, and this is designed to bind to one of those two strands. So as the primer is being extended, the polymerase is going to nudge this out of the way. So it's not going to break it down like in TACMAN. It's going to nudge it out of the way. So it nudges this out of the way, nudges it, nudges it, nudges it, nudges it, and pretty soon this is completely off the DNA, and this strand of DNA is double-stranded, and this strand of DNA is double-stranded, and when it's completely out of the way, boom, it looks like that again. So at the end of the extension phase, it again looks like this. So you need to tell your equipment, hey, make sure you're measuring this at the beginning of the annealing phase so we can say how much of our target do we have, because this is directly measuring how much target this is able to bind to. And the more target it's able to bind to, the more the reporter is far away from the quencher, and so the more a particular wavelength of light you're going to be able to measure. Any questions yet?
Can you go back to TACMAN?
Can I go back to what? TACMAN?
Yeah, I'm just confused as to how it disassociates from the strand.
Okay. So in TACMAN here, you have your, where did I put it down? So in TACMAN, you have your double-stranded DNA. So here's your double-stranded DNA. Here's one primer here. Here's one primer here. And you have a polymerase during the extension phase. And during the extension phase, it's coming along here and it's extending the primer. And as it reaches this extra primer that you've added in here now, it starts to degrade it. So unlike molecular beacons, it's not just nudging it out of the way, it's actually degrading it. So it's degrading it into single nucleotides. so that the reporter and the quencher are permanently separated. So you're getting higher and higher and higher results every time, unlike molecular beacons, which will go up the more target you have, but then at the end of the sequence, when it binds back to itself, the emitted wavelength will drop.
Okay, so it's the extension of the strand that causes it to disassociate.
It's the 5' to 3' exonuclease function of the Taq polymerase. As the Taq polymerase is extending the strand, that starts chewing up the extra primer that you have in there at your target sequence. More questions? You all okay with this idea? Okay, so then the question is, how are you going to be able to, in some situations, let's back off on this a little bit, unzoom it. In some situations, how are you going to be able to use melting temperatures to do multiplexing, particularly in cases of using cyber green? So when you have a known melting temperature, you have a certain sequence, as I said, there's programs where you can say, okay, if this is the sequence, here's going to be the melting temperature. As your DNA target, because your target's going to be much shorter than your template strand. So remember in the PCR lecture, we saw that the template strand can be, your entire DNA genome can be very, very long, but you're going to have primers in a much shorter area. So you have one primer here, and you're just basically going to end up amplifying this really short piece. So as you are getting your target sequence, the equipment can put in a melting temperature. And so, a melting temperature step. So for your target sequence, it will, as it's going into the denaturation step, it's saying how much of this is double-stranded. And this is measuring the change. This is measuring the change basically in double-stranded to single-stranded. So the higher this is, the bigger the change there is. So when it hits a certain temperature, all of a sudden, boom, you'll see a huge change in double-stranded to single-stranded DNA, and that's going to occur at the melting temperature of whatever you are amplifying. So if you have a mispriming event occur, so let's say this is a mispriming event that occurred right here, and this is what your target should be, but maybe not all of your target was correct, and you had some that was an incorrect sequence, or maybe you have two targets, for example, with different amounts. So as you are increasing the temperature going towards the denaturation step, all of a sudden you'll hit a particular temperature where you'll see some of your double-stranded become single-stranded, and that'll be a little bump right there. And then another, at a higher temperature, you can see more of your single-stranded become double-stranded. So if you are going for, say, two different, let's say you're doing cyber green, and you're trying to do multiplexing, you're trying to do two things at the same time in the same tube. And remember, cyber green can only say how much double-stranded do I have. It cannot say, which sequence is this. So you can have an incorrect sequence, or maybe you have a shorter strand and a longer strand that's being amplified. As in the case, for those of you you've already had the STR lecture where you learned how the police department has different sizes of target sequences, and so those different sizes of target sequences are going to have different melting temperatures. So if you needed to, you can tell a shorter strand from a longer strand, for example. Now, this would not be used in police work because 4 bases is not enough to really say, is there going to be a huge difference here? But if you were doing multiplexing of a larger target versus a smaller target, you could say, okay, I've got some of this size and some of this size, and you know what the melting temperatures are, because based on your target sequence, there are, again, these programs that can calculate it for you. So that's what you would do in the real world if you needed to do multiplexing on something with CyberGreen. You'd use the idea of melting temperatures that hopefully you learned something about in 560, maybe? I don't know whether they teach that anymore. I know they did when I had biochem. Okay, any questions on any of that? On your last DNA exam, there is going to be a question on real-time PCR. If there's no questions, I'm done. Get out 10 minutes early. 8 minutes early. Yeah?
So basically for the exam, there's going to be material and everything that we've seen so far?
Yeah, not so far. It's in the DNA section.
Of the DNA.
Yeah, it's not cumulative. So everything that you've done in the DNA section. So that includes the GSTSH2 experiment, it includes the plasmid, it includes the STR experiment. There's going to be a question on sequencing. There's going to be and next week you're getting next gen sequencing. So I think that's the last lecture that you get is on next gen sequencing. So there's going to be a question on tissue culture and stem cells. You had a lot of material, and it's not like incredibly complex. It's a fairly, like the question I asked on tissue culture and stem cells, it's basically going to be something I specifically said in my lecture. And so a lot of the questions are like that. It's things I specifically said in my lecture. Yeah.
Are there any other calculations that we need to know?
Oh, there's always calculations. There will be a calculation question. You do calculations in the plasmid prep, right? Yeah. You do calculations in the STR experiment. So any calculation that you do is fair game. Yeah.
This is a question about the different types of PCR in real-time PCR. So with the last one, with the loop, would that be reusable then?
Yes,
Compared to the
good question, yeah. So, well, you wouldn't reuse it from one reaction to the next.
In the same reaction?
But in the same reaction, it's usable, yeah.
And then for the TACMAG, will you have to keep replacing it for that same reaction then?
No, usually you're putting enough in that you're going to have enough to degrade for your entire reaction. When you set this up with a company, they give you amounts. They say, they'll give you a protocol that says add this, add that. So there's, amazingly enough, when you start working in biotech, if you're doing some of the basic work in biotech, usually somebody else is figuring that all out for you. But if you want to progress in your job, you're going to have to be the one that knows how to how to do the math and how to do the calculations. But when you're starting as a first job, probably someone else is doing the calculations because they don't want you to mess it up. I'm done. If you don't have any questions.